Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Praise be to Allah Subhanahu wa Ta'ala who has given us the opportunity to be able to share knowledge. Introducing myself, my name is Wahyu Surakusumah, lecturer in Biology Study Program. My research field on bioremediation. On this occasion, I represent my colleague, Ibu Periswati and Hoironisa, to present the result of research at the 2021 MSA seminar. Our presentation is entitled The Role and Dynamic of fungi population as a remediator in used motor vehicle oil biodegradation. Along with the development of transportation technology, vehicle production continues. So to an increasing trend, and this cause of the production of motor vehicle oil to continue to increase. Used motor vehicle oil is derivative of petroleum which contain polyaromatic hydrocarbon, chlorine hydrocarbon, and heavy metal, which are carcinogenic and neurotoxic to human, and not only harmful to human. If these waste pollute the environment, can cause disturbance as reported by oil well. The impact of used vehicle oil pollution in the environment can also harm the ecosystem, such as physiological disorder in plant, like decreasing the number in the form of chlorophyll and protein, which can cause a decrease in the photosynthetic ability of phytoplankton. To overcome the problem of the pollution, there are several alternative technologies that can be used, one of which is bioremediation. According to Hidayat, bioremediation is an alternative technique that is economical, environmentally friendly by involving bacteria, fungi, and plants, which has been proven to reduce hydrocarbon pollutant. Physical and chemical techniques are relatively expensive and sometimes produce toxic intermediate. Many research on this bioremediation of used motor vehicle oil have been carried out and have identified bacteria that have the potential for remediation of used motor vehicle oil. As reported by Basuki, 7 bacteria have been identified as bacteria that have the potential to remedy used motor vehicle oil, including Acinetobacter, Achromobacter, Atrobacter, Lactobacterium, Pseudomonas, Bacillus, and Alicaligenes, which are known as hydrocarbonoplastic bacteria. Apart from bacteria, fungi have also been identified that by a rule in remediation of used motor vehicle oil pollutant reported by John Okafor. Several fungi have been identified as having the potential to degrade used motor vehicle oil, used motor vehicle oil pollutant, including Aspergillus, Penicillium, Fusarium, Amphorpotheca, Phaeolomyces, and Talaromyces, and the species of Candida, Yaroia, and Picia. Fungi have an advantage over bacteria because they can produce enzymes that can react with several compounds at once, and fungi can live in extreme conditions. Many research on the bioremediation of used motor vehicle oil have been carried out, but focus on identifying bioremediator organisms and their efficiency. But how the biodegradation process is facilitated by fungi has not been widely studied. Based on this background, we conducted research with the aim to identify the potential of fungi and the role of fungi in biodegradation process of used motor vehicle oil. The research The research method in we divide become 3 stages, namely the isolation of fungi using standard port method, the oil degrading fungus test in this study use a single fungal culture and the fungal consortium consisting of Aspergillus, Penicillium, Trichoderma, consortium fungi, consortium 1 Aspergillus and Penicillium, consortium 2 Aspergillus and Trichoderma, consortium 3 Penicillium with Trichoderma, and consortium 4 Aspergillus, Penicillium, and Trichoderma. The second step is screening of hydrocarbon degrading fungal isolate using the yes mold number method. The isolate were planted into medium, namely medium without used motor vehicle oil and medium with the addition of 10% used motor vehicle oil. The next step is degradation study and hydrocarbon analysis using the gravimetry method to determine the removal efficiency and analyzing change in the relative group area on the GCMS group to identify the change in hydrocarbon composition. In data analysis, we analyze the dynamic of fungi growth. If the growth of fungi in medium that was given used oil was better, indicating that the fungus has the potential as a mediator because it is tolerant of pollutant and is able to utilize pollutant as a carbon source. Utilization of pollutant as carbon source can be verified by the gravimetric method to show what percentage of the decrease in pollutant concentration due to biodegradation carried out by fungi, while the GSM curve can show change in hydrocarbon composition, whether biodegradation occur, so that there is a change in hydrocarbon compound for long chain to shorter chains. To identify the potential of fungi as bioremediator, the first can we analyze based on dynamic of fungi population culture on the medium without pollutant and the media added with 10% used motor vehicle oil. The results show 3 pure isolate of fungi, genus Aspergillus, Penicillium, Trichoderma, and mixed isolate of the genus Aspergillus with Trichoderma and genus Penicillium with Trichoderma on selected media with added motor vehicle oil pollutant was better than grow on selected media without added pollutant. This indicate that the fungus has the potential as a mediator of used motor vehicle oil pollutant and indicate the fact that the isolated fungi are able to use hydrocarbon as substrate for growth by possible releasing extracellular enzyme and acid. They are able to break down stubborn hydrocarbon molecule by disabling long chain of hydrogen and carbon. The result from the other 2 cultures show that the mixed culture of genera Aspergillus, Penicillium, and Trichoderma, and mixed culture of genus Aspergillus with Penicillium, which show no better growth that the culture grown on selected media that was not added with used motor vehicle oil pollutant. This indicates that phenomenon of antagonistic properties between fungal strain in the consortium, so that the growth of the fungal consortium has decreased due to competition of for nutrient. To verify the potential of fungi as a mediator, we can identify based on indicator of the ability to reduce the concentration of pollutant in the medium. The results show that all cultures were able to reduce pollutant concentration from 39.2% to 68.23% in 15 days. To identify change in the hydrocarbon composition of pollutant, we can analyze based on change in the relative area of the GCMS curve. If there is a reduction in the relative area after treatment, it indicates the biodegradation of the component has changed into another hydrocarbon component that is simpler or has a shorter chain. If the relative area on the GCMS curve after treatment indicate increasing, this is indicate the concentration of hydrocarbon component increase due to the biodegradation process of the complex hydrocarbon compound into hydrocarbon component. The result of the change in the medium grow by Aspergillus show this isolate can break down the carbon chain produced in this used oil sample from complex molecule into simple one. Compound that undergo biodegradation are given a red box. It is assumed that the compound that have been degraded or broken down by the fungal isolate will produce the increasing relative area of another compound, and also produce a new two new compound. Change in the hydrocarbon component in the penicillium culture so that 5 component marked with a red box experience a decrease in relative area, indicating that the 5 compounds were biodegraded, while the other 7 compounds experienced an increase in relative area, which indicate an increase in concentration due to biodegradation of complex compound into 7 compound. Change is in the hydrocarbon compound in trichoderma culture, so that 5 component marked with a red box has experienced a decrease in relative area, indicated that the 5 component were biodegraded, while the other 9 component experience in increasing in relative area, indicating increasing in concentration due to biodegradation of complex compound into 9 hydrocarbon compound. Change is in the hydrocarbon compound in medium overgrow with Aspergillus and Penicillium, so that 3 compounds marked with a red box experience a decrease in relative area, indicating that the 3 compounds were biodegraded, while the other 10 compounds experience an increase in relative area, indicating an increasing in concentration due to biodegradation of complex compound into the 10 hydrocarbon compound. Change in the hydrocarbon compound in medium overgrow with Aspergillus and Trichoderma, so that 4 compound marked with a red box experience a decrease in relative area, which indicate the 4 compound were biodegraded, while the other 8 compound experience an increasing in relative area, which indicated an increasing in concentration due to biodegradation of complex compound into 8 hydrocarbon compound. Change in hydrocarbon compound in medium overgrow with penicillium with and trichoderma, so that 4 compound marked with a red box experience a decrease in relative area, indicating that core compound were biodegraded, while the other 8 compound experienced an increase in relative area, which indicated an increase in concentration due to biodegradation of complex compound. Change in hydrocarbon compound in medium overgrowth with aspergillus, penicillium, and trichoderma show that two compound marked with a red box experience a decrease in relative area, indicating that two compound were biodegraded, while the other 8 compound experience in increasing in relative area, indicating an increase in concentration due to biodegradation of complex compound into the 8 hydrocarbon compound. After analyzing the research data that has been described previously, it can be concluded Aspergillus, Penicillium, Trichoderma genera have potential as bioremediator for use motor vehicle oil. This potential is so by being able to grow on selective media that is given 10% use motor vehicle oil and can reduce the pollutant content by 63.28% to 39.2%. Fungus and consortium has a different role in the bioremediation process of hydrocarbon compound contained in used motor vehicle. That's the result of our research. Thank you for your attention. Hopefully useful. Alhamdulillah. Assalamualaikum warahmatullahi wabarakatuh.